Right now, when this liposome is injected, the liposome can fuse with the cell membrane because of the properties are similar with the cell membrane, and the liposome can then uh, release this messenger RNA into the cells. Right. So first, construct the special liposome and put inside this messenger RNA. The active mRNA enters the body cell of the patient. Now it enters. These body cells secrete this protein. Now, if I ask how these body cells secrete the proteins, it's a straightforward because this messenger RNA can enter into this ribosome. Let's say this is a ribosome, and the messenger RNA can now move into the ribosome. Then the ribosome can produce the protein by translation. Now, actually, this is the new type of vaccine because if you remember during the COVID-19, some of the vaccine like Pfizer vaccine, Pfizer, I think also AstraZeneca, these two vaccines, they use this technology. So when this technology used, it is quite new actually, this kind of vaccine. And some scientists are a little bit worried about if they take this RNA, because COVID-19 has a RNA molecule. COVID-19 is a RNA virus, and they take piece of RNA from the COVID-19, and then they inject to these liposomes, and then liposome inject to the human. Then human cell get this virus RNA. That was actually the fear. If virus RNA going into the cells, they had a little bit of fear because you don't know what are the other things can happen. The RNA can serve as a messenger RNA, can make proteins, and these are virus proteins. So when the virus proteins are produced, body can make an immune response to this protein. So the example what I told, antibodies can make. The antibodies are specific to this protein, and antibodies can identify the virus. They also had the question if there are any other things this virus RNA can do in human cells. And then other vaccine like the Sinopharm vaccine, they use whole virus. The virus is completely inactivated. That means their mRNA, the RNA molecules are not active. This virus RNA cannot, but they are proteins. Virus proteins there, but RNA is completely inactive. They cannot do anything. But this case, RNA is active. RNA produce protein by human cell. The virus protein recognized by our immune system, and it makes immune response. So then immune response, we have everything ready. In case virus comes, immune system can quickly start the process, so we don't get the disease. Right? So this process is given here, how it made. Now the question is, I think first mathematical question. The diameter of one spherical liposome was 75 nanometer. Calculate the volume of this liposome using the formula volume equal to this 5R I think it's 3, right? Power of 3, yeah. Right, can you quickly calculate the volume of this liposome? Okay, we have to take this phi as a 3.14 is a standard value, and this one is a diameter. So we have to take the radius 37.5. So if you calculate this value, we get at 3.14 multiplied by, and again multiplied by 4 divided by 3.
I think it is 22/7.
It's okay. That's what the same. Yeah. Right, we get this value, right?
Yes.
So if you want to express this, it's a nanometer. So we want to express in a standard form. We can write it 1.2 1.2 power of 10 to the power 9 nanometer standard form. Right, the next question is active mRNA is formed after post-transcriptional modification of the pre-mRNA. So it's what we learned today. If this is a DNA has exon and introns. Then the DNA copied into the pre-mRNA, and the pre-mRNA length is same as DNA length. It also has exons and introns. But then splicing, the introns are removed and exon joined. After that, we get this is the active mRNA. The question is asking compare and contrast the structure of pre-mRNA and active RNA. Right, the compare and contrast type question, the format is first you have to write the similarities. Then after that the differences. So here what we can write the similarities pre-mRNA and active mRNA both are single strands. So you can add maybe to the same place consist of adenine, uracil, cytosine, and guanine nucleotide. So both, there's no different like the DNA and RNA. They have only these 4 nucleotides. Right, I think always we get more differences than similarities. So then we can write the differences. Under differences, we can list pre-mRNA is longer whereas active mRNA is a shorter, right? So pre-mRNA is longer, whereas active mRNA is shorter. Then we can write pre-mRNA as both introns and exons, but whereas active mRNA has only exons. Now already you have 3 points to get 3 marks. For the last one, pre-mRNA and active mRNA, we can write the pre-mRNA, the active mRNA may have less number of exons, because when it splicing, they can join less number, but the pre-mRNA has all the exons, but active mRNA may have a less number of mRNA. Because of the splicing. Okay, I think you have an idea what other question can be asked from RNA splicing. Right, the next lesson is phenotype, genotype, and the environment, effect of environment, right? Phenotype and genotype. Okay, what is the idea of this phenotype? Phenotype is what we can see, the physical appearance. What is the outcome? So this outcome is basically determined by the genotype. So genotype can decide the outcome. Then is it completely the genotype decide the outcome, or are there any other factor can change the outcome? And certain cases we can also understand environment can also change. Environmental factors can also change the outcome. So that is the environment influence, that is the gene influence. But you know, gene is the main factor which decide the phenotype. But there is a small amount of contribution from the environment. And when we talk about the environment, it is only for some phenotype, but not for other phenotype, right? So many environment has no effect. Many phenotype. I think I take other. It's better. Like many. Many phenotypes, no effect. The many phenotype has no effect. Only some phenotype, there is an effect from the environment. Right? Now let's see what would be the effect from the environment, right? Can we conclude that if we look like this, if we look at people in different countries, they have a different shapes, color, and height. Is it genotype or is it phenotype? Is it genetic or environment, right? So now, when it comes to the conclusion, let's say, you can come to this idea, right? So let's these two person, they are identical twins. They have no genetic difference. They are genetically same. And then one person is going to another country and he's living there. And another person is going to another country is living there. Maybe after 10 years, they're going to meet each other. So let's see example. Now this person is going to Sri Lanka. Other person is going to Finland. Right? So probably it is happening, when some people, they don't have children, they take children, they adopt children. So mostly if the family who has some problem with raising them, and they have two identical twins, it's two kids raising at the same time, it's difficult. So there is an incident we have seen. One is giving to another family, and the early childhood, they are separated, and now they are going to meet each other maybe after 20 years. Now you can imagine the picture of these two adults, like the boys or girls, after 20 years. Is there any changes or no change at all?
How would that behavior?
Okay, we can take that consideration, the behaviors. So if we think about the behaviors, because different environment, when they have different influence, behaviorally they are different. Their behaviors are different. But again, question is, we don't know 100% yet whether behaviors like things are controlled by the genes, and which kind of a genes, and how it controls, and where are these genes are a little bit unclear at the moment. But we have some kind of information. It come up next lesson on the epigenetics, how environment changed the DNA molecules. But if you want to understand the lesson, let's take the height of the person. We take one character, the height, as a phenotype. Height, weight, height and weight. We should see some difference, right? Because of the nutrition. So the nutrition can make these changes. So then we understand there may be some kind of influence from the environment, and maybe skin color. Skin color may be different. So then we can understand there are some changes from the environment, but many not. Now we summarize everything into this piece of information. What is it? The variations, the differences. There are two types. One is continuous, other one is discontinuous. Now what is discontinuous? The tongue rolling ability. Some people can roll the tongues, some people they can't. Then if we make a survey, how many people can roll the tongue and how many cannot, so we come up with this kind of a graph. Yes, No. For example, yes, no. So we get a bar graph, only two columns, yes or no. So these are discontinuous variation because they don't have many different patterns. But if you take height of the people, weight or height, so now you can see we can get the short and the tallest. So we can see many different centimeter level or millimeter level differences. So then we can make a graph like this. So this is the height. So let's say how many people, let's say example like 4.9 meter. Like that we can label this graph, maybe this is 6 meter. Like that we can label and we can place, okay, only a less number of people shorter. Like this, majority of the people in the average height. For this kind of a thing is called continuous variation. The continuous variation is it is actually the genotype and the environment. This is only the genotype. Yes, that's how we can clearly make a conclusion. The differences, the phenotype differences are two types, continuous and discontinuous. So can you draw these two graphs, sketch two graphs on the notes section, discontinuous variation, example tongue rolling. And continuous variation, the graph is like this, it is height. Done? Is this clear? Right now, next lesson Yeah, next lesson, it's similar to this one, but I explain it. The identical twin study. So actually scientists, they have done a study and they select the identical twins, and from the identical twins, when they have a different environment, they check all these differences and they listed. So when they listed certain things like height and weight, they see very small differences in identical twins. Right, then how we can explain environment make these changes, right? Especially like weight, you can understand if there are food is too much and food is nutritious, then they eat a lot, then they get a weight. But height is slightly different. Nutrition does, but increase the height, it's mostly genetic.
It's sometimes when you do specific exercises.
Yeah, that's again environment we can say. Exercise can slightly increase the height. But I will show you another picture which can be not taken one by one. Height or weight or these things, just a little bit of a summary of the picture. One is alright. This gives some idea, a little bit of summary of this environment, right? Now let's assume these are identical twins. So you can see, looking at this picture, that this boy is drinking some kind of artificial, like, just give you an idea. We are not going to evaluate the fresh juice and artificial juice, right? It's just some kind of background. Like, see, he's very untidy. The bed is not really making. He's not really focused, and he's not really doing well. And you see it's not a bright. It's a very cloudy and dark. Like, just gives. And if you look at this side, you can see he's more colorful and fresh juice drinking, nicely made the bed, and then the surrounding is nicely, he's keeping clean, and the bright sun, sunlight, fresh air, like all these things. Two different environments. So if these two different environments, they raise, and at the end you can see at the same age, this person ended up at hospital. So he's a more bald hair and very weak and very old type. This person still, you see, fit, doing exercise, and still very colorful and bright and healthy, like all these things we can see. So this is some kind of a general idea, not very scientific, but just to get an idea. If identical twins, they have two different environments, the outcome is different. Now I think if we try to understand science one hand, if we try to understand religion in one hand, we can sometimes see some places the science and some religions has some kind of explanation. And I think if we take Buddhism and the Christianism, like these religions, mostly the leaders of these religions, they said what you get returned, what you harvest, the Jesus said. And Buddha said that if you do good, good will come. If you do bad, bad will come. Outcome. And these days we are seeing it very clearly, right? We have a good example these days. What if you do bad, the bad will come. Then how can this go into the DNA? And the second question, we have some belief that if the parents, grandparents, the generation, they have some kind of environment affecting the children. For example, we sometimes we say that look at the family what they get, what their grandfather has done, these things. And even that time, his grandfather is like this, he did this kind of a thing. Now they have to suffer because it's bad thing. So it's going to generations. Now, actually, these things we observe frequently, and we have so much of evidence. And even though we are not documented, but we have seen many times these kind of things in the society. That means is our lifestyle, our surrounding, and our emotions, our behaviors, or maybe our intentions. Because if you always think bad to do, some bad thinking and bad action to the society, people, family, or friends like this, we are in a different set of mentality. Opposite is we try to help each other. We try to do good always. If we done something wrong, we can excuse, we can make the corrections like this. Two things existing. Then how can these kind of a thing written on the genes, right? I think that's a little bit going beyond what we know, but we can't explain everything. But there's another picture I can show like this, which can a little bit give some idea for you if I show this picture. Right now, let's see this grandmother, she's smoking. When she is smoking, the chemicals in the cigarettes can affect the egg cells, the ovum of this baby. She has a baby, and now her baby's egg cells. I told you that during the mammalian structure of the gametes, if you take the female side, the egg cells they start to undergo meiosis, and the set of egg cell like 800,000, mostly like around 800,000 preserved, and then they are stage of meiosis, certain stage of meiosis arrested. So now the chemical can affect these cells. When the chemical is affected the cell, it's actually grandmother did this. But now this baby is born here, I see, and this baby is born and become an adult mother. Now she is pregnant. Now when she is pregnant, the egg cells are not healthy because it's done by the grandmother. The bad thing done by the grandmother because the chemical in the cigarette. Now who is suffering now this after this baby is born? She has some kind of a genetic defect. And who did? Grandmother. So it's going to generation. So then how it is going to generation, we can understand the genetic information is going from one generation to next generation. I think we ended up, we will see the mechanism next week. Okay, it is done the class for today. What is this mechanism, how we change the DNA, we will see the next class, right? Okay, then see you next time.
Thank you, sir.
Okay, bye.